and then after break, we'll talk about fertilization. That won't take very long, probably about 30 minutes. This won't take very long either. Fertilization will probably be about a 30 to 45-minute discussion. And then we have 2 things to accomplish in lab, in addition to going over more of the female reproductive models. The sea urchins came yesterday. Yesterday, because they weren't here on Tuesday. Normally in the wintertime, we get our best sea urchins because they come from Monterey Bay. These had to fly in from Florida. And some of the daddies weren't very happy, and the eggs swelled up and ruptured on their own. But maybe that was just one. That's why I was a little late opening up the door. I was talking to our lab tech. And the lab tech that was supposed to take care of that was out sick. So fortunately, the other lab tech used to do this and loves doing it. So she got all of the sea urchins ready. And we'll keep our fingers crossed. So we have 2 and 4 cell stages to look at. She got some fertilized yesterday. And we'll keep our fingers crossed that they will be happy and interact with each other appropriately so that you guys can see it. It costs almost $200 to get those sea urchins in. And we used to limit that when we were everybody had to do the labs on the same day so that we could handle it. And so the decision was made that this was such a good lab that we could do it whenever we wanted to, just so that we could do it. So hopefully we'll get that to work. All right, so as you recall, we finished up lecture. Any questions on what we talked about on Tuesday?
Did you go over the sensory pathway for?
No, because it's the same as the male.
Okay.
And thanks for reminding me. I did finish the changes and additions I was going to make last night. So I put 2 things. I uploaded 2 things onto D2L. There's a modified male reproductive series of PowerPoints. I added a few pictures, but it wasn't complete from what I had used in lecture. So the old one's still up there, but I added, modified it, kind of reorganized it a little bit more to match what lecture was. And then I also, remember the diagrams I drew in the board for the nerve pathways? We were over in the other room. Mm-hmm. So I redrew those with the computer. I thought I would draw it on hand and then scan it, but our scanner doesn't do it in color. So there's several maps. I integrated this into the modified lecture notes, but if you just wanted this. So I just drew out a map of what was what and labeled the structures. And then, as the second lecture, I didn't go through all this again with them. And so then I went through, so then this slide is erection, so I've added, let me redo that. I had a really hard time recopying it. This is actually ejaculation. So let me redo that. But anyway, that won't make any sense. So this is emission. And ejaculation, I actually put a different slide up there for erection. Okay? But this is what I diagrammed on the board with you guys in the other classroom. I think the one that's in the reproductive, the modified reproductive one is correct. Just double check that. Copying the slide and then moving it over, everything got centered and anyway, just a little bit more difficult than it should have been. So this one is erection with parasympathetic. So that's correct in the modified version. And then emission. So I'll double check on that and make sure those are. It was after midnight when I finished this. So. We've got it done earlier if I hadn't been sick. Okay. So, remember we were talking about the hormonal changes, and just to review with you over here on the side, LH is going to theca interna cells. And FSH is going to follicular cells. And theca interna cells are taking cholesterol and converting it to androstenedione. And that is being converted to testosterone. So again, same thing that we saw in the male. In the follicular cells, both of these are being converted to estradiols, which are forms of estrogen. Okay. So we've gone over that pathway on Tuesday and compared male and female. So what we're seeing then is if we look at this here, at the level, this is our menstrual cycle. And the menstrual cycle is standardized at 28 days. So the beginning of the cycle is considered to be the beginning of menses. It's really the end, but it's just easier for women to keep track of the beginning of the period. Sometimes some days are spotty and it kind of tapers off, and it's just a little more difficult to determine. So menses begins. And day 14 is ovulation. Okay. So looking here, you can see that towards the end of the cycle, both estrogen and progesterone levels are falling. So what does that do with negative feedback? What's the influence of those two hormones on the hypothalamus? As they fall, what's going to happen to the level of GnRH? It's going to go up. Okay? So we don't put, well, some texts might, we don't put LH and FSH on this same graph because for some reason they're measured in different units. So it's like having pounds versus kilograms. It's just doesn't quite fit, but we'll compare them side to side. So as progesterone and estrogens drop, they cause the hypothalamus to increase GnRH. Okay, and we see then an increase in FSH and LH, and you can see the inhibin levels from the follicular cells are dropping, which allows the anterior pituitary gland to also make FSH. So what is FSH going to do based on what we talked about on Tuesday as far as the follicle development? Remember, we started out with a primary follicle. And how did we describe a primary follicle? Simple, a single layer of simple squamous epithelia. Okay. So we have several oocytes that are going to be binding FSH. Primary follicles with primary oocytes inside them. And those flat squamous appearing cells are going to have a few receptors for FSH. And now the theca interna cells aren't very well developed yet. So it's not like the follicular cells are making a lot of estrogen, but the effect of the FSH is to increase their synthesis organelles. So they're going to increase ribosomal ER to make it the enzymes, and they're going to increase smooth ER and the synthesis of estrogens. So we're going to see, under the influence of FSH, we're going to see these cells increase the number of, actually let me draw this closer to the beginning. We're going to see these cells increase the size of their follicles and increase the number. In actuality, I'm already halfway through here. This is going to be way back here. Okay, so this diagram should be stretched way out. So now with increasing, this is FSH doing this. So now with increasing cells, all right, and increasing receptors, remember we also saw about this stage, we started to see the theca interna cells. So the LH is going to bind to the theca interna cells, and now we start this process. The theca interna cells are taking cholesterol and converting it to the products that need to be modified to estrogens by the follicular cells. So now we start to see an increase in estrogen as we move into the secondary follicle stage. So now we have secondary follicles, but still primary oocytes. So the oogonia that became primary oocytes when this ovary was in fetal development haven't yet divided. Okay, they're just modifying their internal structures. Again, as we move through these stages, these cells increase the number of follicular cells, or we can now call them granulosal cells if you wish, but they're still follicular cells. So we get more and more and more and more and more follicles. What's going to happen to the level of our estrogen? It's going to go up. And so there's a direct relationship between the number of follicular cells or granulosal cells that are present, that's appearing in the blood. Remember, these are still secondary follicles. As long as we don't see any spaces of fluid, it's a secondary follicle. Finally, we start to get sufficient a number of follicular cells making the estrogens that they start to pool. The fluid that's rich in estrogen starts to pool in gaps among the follicular cells. At this time, we also are going to see the zona pellucida, that protein coat that I told you is around the egg. So at first these spaces appear scattered. There'll be 2 or 3 on one side of the egg and 2 or 3 on the other side of the egg. But eventually they will coalesce. So at any time you see a space, whether it's just a little one here, maybe another one over here, this now identified as an antral follicle. Those spaces are referred to as an antrum. What's the stage of our oocyte?
Still primary?
Still primary. Crossing over is going on, okay? And it's moving, the tetrads are moving into the metaphase stage. By now, even though several of these may have started to develop into secondary oocytes, by now only one, usually, is going to be forming the larger antral follicle. And so again, if we look at our estrogen level, let's see what's, it is rising through all of this. Until about 24 hours, just approximately 24 hours before ovulation, it peaks. Now typically, what would we expect for our GnRH level here? It's going to be suppressed because of negative feedback, all right? But this level of peaking at estrogen actually switches and becomes a positive feedback signal. So low levels of estrogen trigger a release of GnRH. High levels of estrogen are going to inhibit it, all right? But this moderate level is going to result in a release of GnRH and a release of a peak of especially LH. So this becomes a positive feedback we have a spike in LH that occurs just approximately 24 hours. This would be ovulation here. We'll see a spike in LH approximately 24 hours after our peak in estrogen, resulting in ovulation. So that's considered to be a response, a positive feedback response to the estrogen. Now, after ovulation, LH levels remain a little bit higher, and so our theca interna cells become modified. They swell up as they increase their amount of smooth ER, and the entire follicular structure undergoes a very characteristic change. Looks very different from what it did prior to ovulation. And now, although we will continue to make some estrogen, the primary hormone after ovulation from the theca interna cells is progesterone. Okay? So I'm going to erase this diagram here just to allow me to continue the picture, but before I do so, in the theca interna cells now, we are making primarily progesterone. Still, follicular cells are making estrogens, but the major hormone after ovulation is progesterone. The major hormone prior to ovulation is estrogen. So, can I erase this now? You have it on the tables and graphs in your PowerPoints. Yeah, picture. I'm leaving the cells up there, just taking the diagram down. So I need a wall in the other room that wraps around it. Actually, I need the whole room to spin. So I can just slide them around. I don't know why it works. It just has the reverse effect. Normally you would expect that spike to lower GnRH levels, but it doesn't. So that particular level, for some reason, it causes the reverse reaction to occur and actually increases DNRH, which results in increases in the pituitary hormone.
At ovulation?
Yeah, well, just prior to. So the spike in estrogen is about 24 hours. And then the spike of LH occurs just barely prior to ovulation. All right, so now we have at this point, this cell ruptures. This structure ruptures. So what we see, this becomes very large and is known as a mature antral follicle. Okay, its appearance, I'm going to move this name over a little bit just to give me room. So at ovulation, this would be proportionately like 4 times this size. Okay, so I'm not going to draw it that large, I'll just kind of show the rupture of it. So we have our oocyte that's being released with its zona pellucida. Remember now, however, where I draw the zona pellucida, it is a secondary oocyte. And so we are going to have a polar body associated with it. What would be the chromosomal pattern of this secondary oocyte? 2 parentheses, yes. Okay. So we have just the sister chromatids. We have the 23 types mixed up between what came from the mother and what came from the father of this cell. But we have 2 copies of each of those 23 in. Okay, so 2 parentheses in. It's not diploid, it's haploid, but double copies. Then around the first polar body, which is also 2 parentheses in, we have the zona pellucida. So this is what I showed you. So, not quite ready for that one. Then we have a few cells that are released. There is a single layer of these granulosal cells that remain attached in this fashion, known as the corona radiata. And then we have a cluster of cells around it. And that's our ovulated egg. Left behind are the theca interna cells. And other follicular cells that ruptured to release the ovulated egg. And that structure that's left behind, remember I said the theca interna cells become quite large. There is no gap in the middle, at least not one that's discernible. The follicular cells also become enlarged, and they would form a structure all the way around this. And this secretory structure is known as the corpus luteum, because the theca interna cells, when they get all that extra lipid for converting the cholesterol that they're going to convert to progesterone, becomes a yellow body known as the corpus luteum. And these red cells are making the progesterone, and the purple cells are making the estrogens. These were the theca interna cells. These were the follicular cells. All right? So these cells are going to make progesterone. These cells are going to make estrogen. So our estrogen level is going to fall and then slightly rise again. And then at the end of ovulation, at the end of the menstrual cycle, will fall. So this is estrogen. In the meantime, remember our progesterone levels were low. They stay low until after ovulation, and they rise and become the highest level. Yes?
What we do at the end, the purple and the red, what is that in relation to the ovulated secondary
The
oocyte?
purple and the red, this?
Yeah.
This structure is this, and then it ruptured and released the secondary oocyte. This ruptured structure got left behind and converts from the typical appearance of just a follicle to this large secretory structure known as the corpus luteum.
Oh.
Okay, so I'll show you some histology slides. It's this ruptures, and then under influence of LH and FSH converts to that structure. Question, Joe?
Does that stay in the uterus or the?
Pardon?
Does that stay in the ovaries or does that follow?
It stays in the ovary. So for every egg that your ovary ovulates, you have a corpus luteum. Now at ovulation, not ovulation, but at the end of that particular cycle, unless pregnancy occurs, this shrinks. Okay? And becomes white instead of yellow, and so it's called a corpus albicans. So for every egg that was ovulated, your ovary has a corpus albicans. Yes?
I have a quick question, I don't know if it's off topic, but
Okay.
for women, estrogen, you get estrogen from the food that you eat, right?
You can. It's called phytoestrogen.
And then for women, for progesterone, is it progesterone or for progesterone?
For what?
Is it progesterone or,
For what?
How do you pronounce it?
Oh, progesterone?
yes, for women, I know they make some birth control, like if, when they produce too much of it, it stops ovulation, right?
Right. So typically, when birth control pills were first started, they were mostly estrogen. That was thought to be the required pill, but that health-wise, an estrogen would work as a birth control pill. It just had, for so many women, it had side effects with breast cancer and with blood clots, women who were sensitive and had histories of blood clots, that it just became too dangerous.
But you can still get estrogen from the food that
You can, and the birth control pills that are used now are more progesterone. So they usually they'll usually a combination of progesterone and estrogen.
How is estrogen developed? It's natural, I know it's in your body, but it's not.
It's formed from the testosterone.
Okay, so estrogen is used in this one.
Yeah. Plant foods, especially soybeans,
Okay.
have phytoestrogens. Okay, and that's why it's such that in Japan, eats a lot of the soybeans, and other Asian countries, that the women have less problems with menopause. Okay, eating for guys, eating a lot of plant estrogens can lower testosterone levels because it works as a negative feedback on their GnRH. So you don't want to, it can have good and bad effects. It's not like you should eat any at all, but having high concentrations of them can affect hormonal levels.
Yeah, why does the progesterone keep being, we're talking about, that's the green, right?
Yeah, so progesterone here isn't as high because the theca interna cells, androstenedione and testosterone, is being transported to the follicular cells, where it's being converted to estrogen. But then the rise in LH becoming a more prominent, we still need the LH for the testosterone and androstenedione, but more of that, the theca cells undergo changes after ovulation, and more of that androstenedione goes to progesterone, or the cholesterol goes directly to progesterone.
So it's not because there's more of them?
Well, yeah, but they undergo a change, they undergo a histological change after ovulation occurs. Okay? So, here's an image that kind of illustrates what I diagrammed on the board without the pretty pictures. So, here's the primary follicle, okay, under